what we have done is we have isolated our protein which has a N-terminal histidine tag using affinity chromatography, and we have used FPLC system to isolate our protein from the other cellular components. There might be other carbohydrates, other proteins and nucleic acids, and those we have separated out and we got our protein. Now, as I have mentioned that you need to check how pure your protein is before you go for any kind of other biochemical assay. So, first we need to do SDS page or gel electrophoresis using SDS page, and we need to check the purity of our protein. So, for that what I have done till now is I have collected the fractions that showed a typical peak in the FPLC system, and I have collected all the fractions starting from 18 to 24. So, there was almost 15 ml of solution, and using this particular Centricon I have centrifuged this those solution and concentrated the sample. So, as I have mentioned earlier as well, this Centricon used as a diafiltration unit. So, basically what it will do is while centrifugation is happening, due to the centrifugal force, the buffer or the soluble water what is there will be pushed out from this filtration unit. So, it has a cut-off membrane and it will be pushed out of this membrane, and also whatever smaller molecules that are not that can be bypassed, that can go through this membrane, will also come out. So, this particular membrane has a molecular cutoff of 3 kilodalton. So, anything which are smaller, like different types of ions and proteins which are smaller in size than our protein, that will also come out from this particular solution. So, it will push out the buffer, the water basically, also the ions and the other smaller molecules. Then from 15 ml we have come down to suppose 2 ml or so. So, invariably what I have done till now is I actually concentrated my sample, and now with this sample I will run a SDS page. So, first let us try with the SDS page. So, now we will make a 12% SDS page, and for that what we have here is the glass plates, and we will assemble these glass plates to cast a gel. So, there is a bigger plate and also a smaller shorter plate, and this needs to be assembled in a gel caster. So, when I assemble this two plate, what it will form is a small gap will be there between these two plates. So, there is a small gap here, and in this small gap I will pour the gel matrix, basically the acrylamide and polyacrylamide gel will be casted here. So, the gel matrix will be here, and I will pour this matrix, and it will form a very fine thin gel for our protein. Now, I will keep this caster in this tray like so. So, now in this tray at the bottom part there is a thick material which will stop any kind of leakage. So, basically it is blocked now, and I can pour whatever matrix or water and the buffer here, and it will not leak out. So, it will give us the time till the acrylamide gets polymerized, and then we can take it out and then we will load our sample. So, now I will make the resolving gel, and for that I will take all the components here in this small beaker. So, first I am taking water. So, this is deionized water. So, to make a small gel like this, this is almost about 10 ml of volume. So, the how much water or how much solution you will take will be provided. So, this you can also go and search in the internet or other literature. It is already mentioned here for a 12% gel how much you need to make. So, the percentage will also vary depending on your protein. So, my protein is almost 15 kilodalton. So, I am making 12% gel. If your protein is smaller than that, you have to go for a higher percentage gel like 15% or 20% or so. And if you have a much bigger protein than this, suppose you are working with 30 kilodalton or 50 kilodalton, then you can go for 10% gel or so. So, right now I am taking 30% acrylamide and bisacrylamide solution. So, this is already a premixed solution. You might also find out that acrylamide and bisacrylamide in a separate container, then you have to make the concentration like 19:1 or 29:1 like that. Now, the resolving gel has a buffer of trace which has a pH of 8.8. So, it is highly alkaline. So, right now I am adding the tris. Now, I will add 10% SDS. So, SDS will give us impart the negative charge to our protein, and also it will denature the protein. So, it will be in a polypeptide form basically. That is why it is called a denaturing gel. When you give SDS, it becomes denaturing. If you do not put SDS, then it will be a native page. But here we are mainly focused on getting the molecular weight of our protein and also the purity of our protein. That is why we are going for a denaturing gel. Now, I will give 10% APS, which will provide the free radicals for polymerization. So, now in this beaker I have water, acrylamide, the tris, buffer, pH 8.8, 10% SDS and 10% APS. At the last I will add the Temet. So, just before you start pouring the gel, you have to add the Temet. If you add the Temet and wait, the gel will polymerize in this beaker and you cannot pour the gel. So, after you add Temet, you have to be very quick and start loading your sample. So, now I will add the Temet. So, Temet has a very distinguishing smell of sulfur. So, it has a bad smell. A very small volume is needed. Just mix quickly. And start loading in this sample. So, leave a small gap where we will cast our stacking gel. Above this, on the top of our dissolving gel, I will add a small volume of isopropanol. So, this will ensure that our gel is at the top section is not in a touch of air. Basically, if there is air, oxygen will scavenge the free radicals and then the polymerization will be disturbed. So, the isopropanol does two work. Basically, it stops the air to get in contact with our gel, and also it gives a clean equilibrated clean level, so that our gel is at the same level all over the place. If you do not have isopropanol, you can just add simple water and then you keep it like that for 15 to 20 minutes till the polymerization is done. Then after that we will remove this isopropanol or water and then we will pour the stacking gel. Now, there are few tips here. If you want a very crisp, very fine band for your proteins, then you can also add glycerol. 1% or 2% glycerol will give make the gel quite good, and the bands, protein bands will be formed very crisp. Also, if you really want a good gel, you can also the sample which I have poured right now, the acrylamide and the tris solution, you can degas it before you add the TEMET, degas whole solution, then you add the TEMET and then you cast the gel. Then what will happen is there will be any kind of oxygen that is trapped inside the solution which you are pouring, like the buffers and the water and the acrylamide, that will be out of this whole solution, and the casting or basically the gel polymerization will be very good, and then you can have a very nice gel. So, if your experiment or your gel is very important, you are going for a publication or so, you can you can degas the whole solution, then pour the gel. Also, you might add some amount of glycerol, 1% or 2% in your solution, and then you can have a very fine band of your gel. And you have to wait 15 minutes or so or 20 minutes before the whole system is polymerized, then only we can start for the stacking gel. So, after almost 15 to 20 minutes, the acrylamide gel will be polymerized and it will be solid. So, now I will remove the alcohol or the isopropanol which I have put on the top of this gel. So, I will just soak it up with the help of a tissue paper. So, just tilt the whole system. Do not deassemble the system before you have casted the whole thing. Now, to clear out the whole propanol, I will just pour some water, and then again I will just tilt the system and just soak it up. So, as you can see, the gel is the resolving gel has been prepared and it is solid, and there is a small gap where we will put the stacking gel. Now, if you feel that your gel is not getting solidified even after a long time, either you wait for more time, or if you feel like you can add some more TEMET and it will help to polymerize the gel quickly. But do not put too much of TEMET before, again if you put like more TEMET, then you might be feeling that while you are casting the gel is getting polymerized, then it will make a very bad gel. So, there should be a balance between the TEMET and the time, but if you want to do quicker, you can add some more TEMET as well. Now, I will start with the casting the stacking gel. Again I will start with water. So, stacking gel will be almost 1.5 ml or so in volume. It is a small thing. It will ensure that all the proteins come at the same front line and starts from the same place. Now, I will add the same acrylamide, but in a very small volume. So, the stacking gel has larger pore size, and in the stacking gel the protein even with the difference of size will run at the similar rate. So, it does not work as a molecular sieve or so. So, pore size are very big, so protein gets to run at the same side. Now, I will add the tris buffer, and here the tris buffer has a pH of around 6.8, and the concentration of the tris is also very low. So, there in the resolving gel the tris was around 1.5 molar tris, pH 8.8, here the tris is 0.5 molar tris and the pH is 6.8. Now, again I will add the APS and the SDS first. So, in this particular denaturing gel there will be always SDS in all the gel, so resolving as well as the stacking. So, I have taken SDS as well as the APS, both are of 10%. Then at last I will put Timid. The moment I put Timid, the gel will start polymerizing. So, again be careful, just before you are ready, you just add the Timid. Do not wait. After adding the Timid, just mix it gently and then pour on the top of this resolving gel. Now, before it starts solidifying, just add the comb which will make the wells where we will load our sample. So, I am using a 10 well comb, just press it in that particular gap and it will make small holes or wells there. There will be some spillage, so it will go up because the comb will display some amount or volume of the stacking gel. Do not worry about it, do not pour little to avoid this spillage, because if you pour little there might be a chance that a bubble gets trapped there, and if a bubble is there inside your gel, the protein there at that particular local environment will not run properly, because again the current or voltage during the polymerization, during the electrophoresis will vary, and if there is a bubble, the whole gel will be bad. So, fill up the stacking gel at the brink of this particular line and then you adjust the comb and put it there before the gel solidifies. Now, you again have to wait for some time, after that the stacking gel gets properly polymerized. So, again we will wait for around 15 to 20 minutes. In the meanwhile, what you can do is you can prepare a sample. So, for SDS page or in case of denaturing gel preparation, what we do is we have a particular loading die. So, you cannot load your sample just as it is. So, you have to have a die. What is the importance of this die is it has first of all it has a dye which is the bromophenol blue. So, it will run very fast in the gel and you can actually see where your sample is basically. So, you can monitor the run during the electrophoresis using this dye. So, it will not interact with your protein, it will not create any trouble during the run or electrophoresis, but it will help you to monitor how long it has been run and when you have to stop the electrophoresis. Also, there are another important ingredients inside this dye or this gel loading dye. So, basically it has glycerol. So, when you have a sample that is a protein, it is quite light. So, basically it is in a buffering system which is mostly made up of water, and when you try to load your sample inside a well, it will float out because it is not heavy or the viscosity is not high. So, it will try to float out. But when you add glycerol, because glycerol is a heavy molecular weight substances, it will make your sample to go inside the well and sit at the bottom of the well. So, the glycerol will help your sample to go at the bottom of the well. If you do not have glycerol, your sample loading will be hampered and your whole protein will come out from the well. Another thing is in this particular denaturing condition, there is also SDS in this dye. So, what during gel preparation or basically sample preparation for the gel, what you have to do is, suppose you have a protein solution. So, I have made the protein and it is in the liquid form. So, it is a white basically a colorless liquid. So, you take small volume of this protein, suppose 4 microliter of this protein, and then you add that particular sample in this gel loading dye. So, this gel loading dye is 4x, that means it is 4 times what you are supposed to use. So, the whole concentration is 4 times more. So, here what you have to do while loading, the dye will be around 1x. So, basically you have to make the adjustment of the concentration by adding your sample and if needed the water or something else. So, you have to make it 1x. So, basically I will take around, suppose if I want to make a 16 microliter of sample for loading, I will take around 12 microliter of my sample and 4 microliter of this loading dye, and then the whole sample volume will be 16 microliter, where the dye concentration will be 1x. So, you have to make it 1x. You cannot make this dye 1x, because then what will happen, you cannot add anything to this particular dye. If you add anything to the 1x dye, then the dye concentration will go down to 1x, and that is not right. So, make it in a 2x, 6x or 4x concentration, so higher concentration, and when you add your sample, then the concentration will be go down to 1x. So, this you have to keep in mind, that you have, when you start loading, you have to make the whole dye as 1x, and then you make the sample like that. So, again I will take 12 microliter of this sample and 4 microliter of this loading dye, and then the whole sample concentration will be 16 microliter and the dye will be 1x. Now, that is not over. Now, you have to boil the sample. So, boiling, what will it do is, it will denature the whole protein system. So, protein has tertiary structures or quaternary structures, and it will break down, because SDS or the sodium dodecyl sulphate, which is an anionic detergent, is present, and it will break down the whole system, and the protein complex will be broken, and the whole protein will be remain as a polypeptide chain basically, and that will ensure that depending on the molecular weight, it will move at a certain rate. So, if you have a protein which is of 20 kilodalton, and if you have a protein which is of 40 kilodalton, the 20 kilodalton will be running almost 2x faster than the 40 kilodalton, because it is of lower molecular weight. But if you have, suppose you do not add the SDS page or SDS detergent, what will happen, that if your protein is folded in a certain conformation, or it might be forming a dimer, or it is making complex with another protein, then that particular complex will have a bigger molecular weight, and you cannot estimate the actual molecular weight of that protein. So, to get the actual molecular weight of a protein, and with respective to a particular molecular marker, you have to put SDS in that dye and also in all the system, so that the protein breaks down in a polypeptide chain. SDS will also ensure that protein is properly negatively charged throughout. So, it does not basically choose or selectively make negative charge, it just randomly it just put a negative charge in all over the place. So, protein will be negative charge, and it will run from negative charged side to positive charged side. So, then what we have done is, we have added the sample and then make this particular sample, and then boiled in either a water bath which has around 100 degree centigrade, or a heat block, dry heat block you can also use, which is around 100 degree centigrade. So, 96 degree centigrade to 100 degree centigrade, you have to boil for almost 10 to 15 minutes, and after that you just spin it down or centrifuge it at the high speed, so that any kind of particulate matter or present in the dye or your sample will be precipitated out, and then basically as a pellet form, it will come out from the solution, and then you add the this solvent in the well, and then you start the running. So, I have boiled my sample for 15 minutes. Another tip is, so suppose you have a whole cell lyser. So, suppose you are taking 1 ml of LB culture, and then you take the pellet, and then you took the pellet and as it is suspended in a buffer, and then you added the dye. So, in that case, what you have to do is, you have to boil for a longer time, because in that particular cell pellet, DNA, RNA and other carbohydrate or other molecules will be present. Then you have to go for a higher temperature or boil for a longer time, so that the DNA, RNA whatever is present will be broken down in a smaller piece, and it will not hamper the gel running. So, if you have a purified sample, the boiling time can be reduced, so 5 to 10 minutes is fine. But if you have taking a cell pellet or crude lyser, then you have to boil it for a longer time, 15 to 20 minutes or 25 minutes, so that everything is broken down, and then you have to centrifuge it, and then you load only the supernatant. Another important thing is, as I have mentioned earlier as well, that if you have a cysteine, single cysteine residue, and there might be a chance that it forms a cysteine dimer, in that case you need to run in a reducing environment. So, for that you can add 1 millimolar of BH BME, like the final concentration of BME will be 1 millimolar in the loading this particular sample. So, basically you just add BME in the sample and then you boil it for some time and then you load it. So, what it will do is the BME is a reducing agent, it will cleave the, or basically don't make the formation of disulfide bond, and it will cleave the disulfide bond and make it the SH only. So, cysteine residues will not form disulfide bond and it will have a reducing agent, and then you can see only a single band. It might be you can see that sometimes when you are running a gel and you are seeing two bands equally, suppose your protein is 15 kilodalton, you are seeing a band at the 15 kilodalton as well as a 30 kilodalton, then might be because of a dimer formation. If you add BME, then only you can see a 15 kilodalton band and that particular dimer formation will be hampered. But suppose you have multiple cysteine and that particular cysteine are important for your folding, then do not proceed during the biochemical assay, do not give this BME. Only when you are running a gel and you want to calculate the molecular weight and see how much pure protein you have, then only you add this BME sample, which is around 1 millimolar at final concentration. So, now we have prepared our sample, we have boiled the sample and the sample is ready. Another thing during loading, what you need is a molecular weight marker. So, this is also called a protein ladder. So, in this particular sample it is provided. So, we have bought it from a manufacturing company. So, what it does is it has a different molecular weight proteins. So, suppose 15, 30, 50 like that, and when you run it, it is already pristine. You can see that particular all this protein, it will form a ladder system, and with respect to this ladder you can actually measure or estimate what is the molecular weight of your protein. So, it is important to run a ladder, otherwise you cannot judge where your protein is coming, because then you do not know at which molecular weight you are getting your protein. So, suppose you have 15 kilodalton protein, then that 15 kilodalton protein should match with this particular molecular weight marker. Otherwise, you will never know at what size, which size or where it is coming in the gel. So, we have taken all these things here. Another thing you will need is a running buffer. So, basically that is SDS page running buffer has trace, glycine and SDS basically. So, the running buffer is also alkaline around pH 8.2 or so, and it also has glycine molecule, and this glycine and trace and the chloride ions will help for the migration of your protein, and also it has SDS because in the denaturing page everything contains SDS to basically denature the protein complex, and if where you have the denatured protein, and you can actually estimate the molecular size of the protein. So, now our stacking gel has been polymerized, and then I will remove this whole system from this caster. Then I will open or take out the gel plate from this casting cassette. And this is another cassette where I will start running my gel. So, the smaller gel part or the smaller plate basically will face inside of this cassette, and then I will put another cassette here or another plate here. Basically, it is a plastic plate to make is a closed vessel kind of thing. So, this is the total cassette which I have formed. So, this side the actual gel is there, and at this side this is another just cassette. If you have another gel, you can run simultaneously two gel here. So, you can put the other gel here. As I am running only one gel, I put a just a block system basically, and this is a buffer chamber. The red denotes the negative line and the black denotes the positive line. So, basically here also there is a red and black marking, and this also have red and black marking, so that you do not confuse yourself, and just put it red towards red and the black towards the black. After you put the whole system like this, you have to fill up the inner chamber. So, this whole cassette system makes an inner chamber, and there is an outer chamber as well. So, first I will put it like this. And the gel running buffer which I have talked previously, it has trace, glycine, tracer, glycine and SDS. So, I will fill up the inner chamber first. So, just wait for a few minutes to see if there is a leakage from the inner chamber. If there is a leakage from the inner chamber, then when you are running the gel, it will start leaking and the inner chamber will get depleted from the buffer, and the whole procedure of the electrophoresis will stop because of the charge distribution. It will be hampered. So, another, I have to fill the outer chamber. There is a marking here, it says 2 gels or 4 gels. So, as I am using a 2 gel cassette, I have to fill the outer chamber till the 2 gels. So, it will ensure that this whole system is properly equilibrated and at the same electric field, the voltage will be similar. So, the connection will be there, otherwise it will be discontinued. So, I have filled the outer chamber as well. Now, there are two school of method which you can follow. You can either remove this foam beforehand and then you can wash your wells, or you can just pour the buffer and then remove your comb. So, it will depend on yourself which you are comfortable with, and just pull at the both side the same time, so that you do not disturb or basically disentangle the combs. So, now you have to see, able to see the combs here. So, there will be around 10 combs. It probably will be difficult in this video, but when you are doing it by hand, you can see the combs, and there you have to load your sample. Now, we are ready to load our sample. So, I am loading different samples. So, basically I am loading one sample which I made before the purification. So, there will be other proteins as a contaminant, and then after purification which will contain only our, which is supposed to contain only our protein, but we have to check if it is at all containing only our protein or not. So, I will make it a small volume. So, I have previously prepared the sample, I have added the loading dye as well as the, I have boiled the whole thing. So, just go down, put the tip at the top part and slowly just release the sample. When you are done, do not release your pipette inside, because then it will take out your sample. Just take it out and then release, and then discard the tip. Because of the glycerol and the dye, you can actually see your sample, otherwise you will not be able to see that. Now, I am adding the ladder, which will be having a different color, and it is already pre-stained. So, while running you can actually see the different protein fragments. I have made few samples from the fractions as well, which we have collected earlier, to check all the fractions have pure protein or not. So, I have only 3-4 samples, there were 10 wells. So, basically what I have done is I did not keep the free wells as it is, I have put some amount of buffer, basically the loading die itself. So, sometimes when you are running SDS page, if you have a free well, what will happen is if there is a nearby protein, it will try to go towards that lane and it will push towards that particular lane, the free lane, and the band will not be properly crisp or a single band, and you might get a weird kind of gel. That is why do not keep any free lane in a SDS page gel, always fill it up with some amount of loading die itself. Now, the loading has been done. You can see all the samples are actually at the bottom part of the gel, so or the wells basically. So, what it ensures that because of the presence of glycerol, it has made the whole sample little bit more weight, the weight is more, and that is why it is at the bottom part of your sample well, and if you do not have glycerol, it will just come out from the well. So, always add glycerol to your sample and then load. Now, I will just put this cap here. So, the red will go to the red and the black will go to the black. Now, I will add the whole thing. So, I will basically start the running using this power pack. So, basically this is a power pack where you can actually regulate the monitor or the voltage basically. So, I have this power pack here. So, there is also red and black marking you can see, and in this particular cord there is also a red and black thing, and so basically the red will go towards the red and the black will go towards the black. And here I can change the, so it is showing 0, that means it is at 0 volt now, and it is not showing anything running or pause or anything. So, I will change the voltage. So, I will make it suppose 100. So, this means it is 100 volt. So, V is on. So, there is a blue or basically yellow light going on. So, the 100 volt, and then I will start the run. So, the basic rule of thumb of what voltage you should run your gel is around like per centimeter you should be at 10 volt. So, if your gel is suppose 10 centimeter big, then you can run it at 100 volt or so. So, first it is good to run at a lower voltage like 90 or 80 like that, while the gel is running or the sample is running at the stacking gel, and then after it went to the resolving gel, you can increase the voltage to 120 or 130 to save some time. So, now that I have started running, you will able to see some bubbles going up here. That means basically the electric field has been generated and it is going from negative to positive side. And if you want, you can do this whole thing in a ice box or in a cold room. So, when you do in a cold room, again the gel will be far better than this, and you can keep the whole system in a ice box outside itself. So, you put just ice all over the place. You have to keep the buffer as well in the ice previously. So, it will be chilled, and when you run in a chilled condition, then gel will be far better. So, if you have an important experiment or important gel which you want, you can run it in a cool environment which is cold, and the buffer will also be cold. And so, basically now it has to be run. So, it will take almost 1 hour or so, and you will be able to see how much it has been run, because there is a marker or the basically a front dye, the bromophenol blue will run, as well as we have loaded a pre-stained marker. So, you can see the marker will also separate out, and you can actually able to see that because it is already pre-stained. You will not be able to see your protein because it does not have the stain. So, after the running we will put it in a stain, and then after the staining and destaining process you are only able to see your protein. Another important aspect of this isolation and purification of protein is to know how much protein you have. So, the amount is also important. So, you need to know how much protein you got in a particular from a single purification protocol. So, the gel will help us to see how pure your protein is, and another thing we will do is to check the absorbance at 280 nanometer to get to know how much protein we have. So, it will be important during developing any kind of assay where you need to know how much protein you are providing in a assay system, and to measure the activity of that protein. protein. So, for the getting the estimation of how much protein we have, we need to check the absorbance. So, protein usually have tryptophan and tyrosine residues, which are the this hydrophobic residues have this phenyl ring, and it absorbs the 280 nanometer, and that can be monitored in our nanodrop system. So, I think previously also we have shown that how the nanodrop works. Here also we will see how we can estimate the amount of protein we have. So, as I have got this protein from this so basically from the fractions I have concentrated my protein, and that is the fraction I am running here in the gel to check the purity, as well as using this particular sample I will see how much protein I have. Now, sometimes you might be working with proteins which do not have any tryptophan or tyrosine residues, then it will not show any absorbance at 280 nanometer. Then what you can go for is a gel estimation. So, from seeing the gel band and how much you loaded a sample in the gel, you have to have a basically sample protein which have a known concentration, and relative to that known concentration you need to estimate or measure how much protein you have. If you do not have any tryptophan tyrosine residues which will be absorbed at 280 nanometer. Another methods which are there, like Lowry assay or Bradford assay, for which will be useful to know the estimation of the protein. So, as here you can see that the sample has already passed the stacking gel, and the pre-stint marker has already started to separate out. You can at least see the red band here. So, I have also increased the voltage. Here I have made it from 100 to 120, or you can make it 130 as well to save some time. Otherwise, you can keep it at 100 and run it for a longer time at the same rate. Otherwise, after the it passes through the stacking gel, you can increase the voltage to save your time. So, we will run till it goes at least halfway through the gel, and then we will stop, and then we will put it in a distain. Now, we will measure the absorbance of our sample at 280 nanometer. So, this is a nanodrop. This is a spectrophotometer basically, but it uses nanoscale solution. So, you do not have to have a bigger volume of your sample, you can just use it in a small volume like 1 microliter, 2 microliter. So, I will switch it on. It will take some time to initialize the machine. So, in this particular nanodrop we have a monitor as well as this disc where the handle is there, and in that particular point we will load our sample. So, before we load our sample we have to make the blank as well. So, in this particular sample our protein is there, but it also contains a buffer system. So, basically the blank will be the same buffer without any kind of protein or our sample. So, here I have used only the same buffer which I have tried to get the purified protein. So, basically it has trace and NaCl. During the purification protocol, the buffer which we have used, it is the same buffer here. So, I have to use the same blank here. Always make sure that in whatever buffer your protein is in, you should use that particular buffer as a blank. So, now it is showing that instrument is initializing, and do not lift the arm during the initializing, that means this arm. So, in that particular arm there is a point where the light is coming through, and there you can get the absorbance. This can also act as a spectrophotometer. So, basically you can add a nanodrop scale solution, as well as you can add a cuvette. There is a point where basically there is a portion where you can add your cuvette and where you can have greater volume of your sample. So, in this particular machine you can show or measure the absorbance at different wavelength of light like 600 nanometer or 540 nanometer or so. But we are mostly going to do here right now is for the protein, and proteins absorbed due to the tryptophan and tyrosine residues at the 280 nanometer. So, now in this particular screen you can see there are different options for you. So, nucleic acid, proteins, OD600, custom and kinetics. So, nucleic acid means DNA or RNA. You can also measure DNA RNA at the particular 260 nanometer wavelength. you can measure OD600. Basically, when we grow a bacterial cell in LB media or so, the bacterial cell basically have a measure absorbance at OD600. So, that is called optical density. You can see how much bacterial growth has been there. So, when you just loaded the basically normal LB use as a blank, and then when the bacterial growth will be there, the OD600 value will be more, because the bacterial cells will scatter the light and the absorbance will be more. But right now what we are going to do is go for the proteins. So, even in the protein section there are different options here. Here you can see the protein at A280, proteins and labels. That means, if you have a particular specific label which has an emission or absorption spectra, that you can choose. Protein at A205. So, that A205 means absorbance as 205 nanometer. So, if your protein do not have tryptophan or tyrosine residues, the peptide backbone basically has this absorbance at 190 to 195 nanometer scale, but mostly none of the machine measure the absorbance at 190 because of some noise, because mostly at 190 almost all of the substances or molecules absorb. So, then the protein backbone measurement is done at 205, the best nanometer scale at the higher time point, which is around 190. So, at 205 you can measure the absorbance which is due to the protein backbones, but not necessarily it is always useful or helpful, because at this particular times nanometer range, as I have mentioned, a lot of molecules absorbs. Another thing you can see that there are BSA, Bradford and Lowry. As I have mentioned earlier, that you can also go for Bradford and Lowry estimation. So, these are the reagents which you add in your sample, and the sample changes color, and during the changing color, how much color has been changed, you can actually estimate how much protein you have. Also, you can have a BSA. Basically, BSA is a bovine serum albumin. So, BSA is a bovine serum albumin, and there you can have a estimate as a standard protein. You can make a graph using a standard concentration of BSA, and then when you respect to that particular graph where your protein concentration is coming, you can measure that. I will go for protein A280 nanometers time scale. Here I went there and I will select a type, which means I am going for one absorbance equal to 1 mg per ml. And we will go for done. So, now it is giving me instructions here in this particular monitor, like here it is showing that clean both pedestal and load the blank, and the auto blank is on. Basically, I have to clean the pedestal. So, I will just open up the arm, and then first what I need to do, I will take in a clean Kim wipe, I will take small amount of deionized water, and this tube portion I will clean it. So, if there is any particulate matter or anything that are present, any dust particle will be cleaned, and then I will load first the blank. So, as I mentioned, this is a nanodrop machine, so I can load very small volume. So, right now I am loading around 2 microliter of blank. So, this is the blank in which the buffer basically in which my protein is there. Blank does not contain any protein, by the way, it is just the simple buffer. Now, carefully I will load the blank, and then I will lower the arm. Now, as the auto blank was on, it itself started taking the blank, and the next step it will give us the instruction what we have to do. So, here you can see the blank is okay. So, that means there is no noise or any other absorbance is there. Clean both the pedestal and load the sample. So, it is giving me telling me that you again have to clean the pedestal, remove the blank, and then you have to load the sample. So, that I will do right now. So, I will again open up the arm. I will remove the blank from here, and taking a nanodrop, I will just clean it. So, these are very sensitive instrument, try not to make any scratch there. all the spectrophotometers are quite sensitive. If there is any scratch, then the absorbance will be vary. Now I will load my sample. Again I am taking 2 microliter of the sample and carefully I will load. Try not to make any bubble in that particular small sample, otherwise the absorption will be different, and then I will lower the blank. Now it is showing me a particular value, that is here you can see mg per ml, it is showing me 9.796. So, do not trust this particular mg per ml, it means that at particular one absorbance is coming from 1 mg of protein, but that is actually standardized for a known protein, that is mostly BSA, but our protein is different. So, take this value, that is 8280. Now, 8280 value shows 9.80. Now, there is another value 8260 by 280. So, 8260 by 280 basically shows if you have any particular DNA or RNA contamination, because as I have mentioned that DNA, RNA, all the nucleic acid absorbs at the 260 nanometer wavelength, and if you have a higher value, like here it is 0.6, it is fine. If you have suppose 1.2 or 1.8 like that, that means DNA contamination is there. If it is go beyond 2, that means protein RNA contamination will be there. So, these values is okay. So, we have only our protein and there is no DNA or RNA contamination. And since we got this A280 value 9.8, what does it means? It does not tell us how much protein we have. So, for that what we need to know is the extinction coefficient of our protein. So, the extinction coefficient is basically you can have it if you provide the protein sequence in the ProtParam software, and the website you go and ProtParam software, you just upload your protein sequence and it will calculate the extinction coefficient of the sample of your protein. Here I know my absorption coefficient is around 14.45 millimolar per centimeter. So, that is given in the ProtParam. So, now what I will do is I will just divide this particular A280 value. So, that is 9.80 and I will divide by the molar extinction coefficient. So, the molar extinction coefficient as I mentioned is 14,450. And then if I divide that, I will have in a molar range or the how much molar protein I have. Then if I multiply that by 1000, I will have how much millimolar protein I have. So, now after doing this calculation, so just dividing this 9.8 by 14,450 and then multiplying by 100,000, I am getting around 0.67 millimolar of protein. So, that means in this particular volume of sample, it is around 1 ml or so, I am getting around 0.67 millimolar of protein. So, that is good because 0.67 millimolar is quite good. Another thing, if you want to calculate how much mg per ml protein you have, in that particular ProtParam file you will see there is another option or given that how much absorbance you will get for 1 mg per ml protein. So, in my case it give us that 1.7 absorbance or A280 value will come for 1 mg per ml. So, now you can easily calculate how much mg per ml protein you have in a particular sample, and then how much ml protein you have, you can calculate the total amount of protein you got from a single purification protocol. So, by doing this we can actually calculate how much protein we have, and then it will help you to establish any kind of biological assay system where you need to know how much protein you are giving in a particular experiment. And also if you are not happy with the protein you got in the sample during the protocol, you can always change or modify your protocol and then try to get as much protein you have. Now, if this particular experiment is done, so I am going to end this experiment. Again it will tell me what to do. So, I have to clean the sample area. So, I am just opening this. Again I will take a tissue paper and soak it with just water just clean it. Do not be rough with this, do not pour any harsh chemicals here, and then lower this area and touch the cut. So, my job is done here. So, I will just close this machine. Now, you can see that the gel has almost run, and we can see that by observing the marker. So, the ladder or the protein marker has separated out. You can see the different fragments of that protein. As we are using crescent marker, we are able to see that, but in the this particular part we cannot see anything because our proteins are not stained previously. We have to go for the staining after the running is done. So, now we can stop it because I am okay with the run right now. So, I will just stop the voltage, remove the whole system. Now, I will dismantle. So, you can see the protein marker has quite separated now. So, that means it has these different fragments, and we already know what is the molecular weight of those fragments. And this is basically the front dye which is running. This is the bromophenol blue which was there in the loading dye. Now, remove the buffer and we will dismantle this whole system. Put it here. So, this is basically the plate or the gel which we were running. Now, I will try to separate out the gel from this plate. So, this is basically a gel staining tray which we got here, a box basically, and this is basically a separator which was already provided with the whole gel casting tray, and gently we will try to lift up this small plate from the bigger one. Now, you can see the gel is stuck within the bigger plate. You may or may not remove this comb or basically the wells. If you are comfortable with it, you can just keep it like that. Then in this staining box, I have taken first a small amount of water, and I will just dip this plate. So, the gel will come out in this box. So, we have to do it. So, now that you can see the gel has come out, I will remove the plate here. So, for storage we need to clean these plates and then keep it. So, I am just washing it once lightly with the water, and then I will pour out the water. Now, I will pour the staining solution. So, this is Chromacid Brilliant Blue which will stain our protein. So, I am just pouring the staining solution here. Closing this, and now we will keep this in a rocking motion for almost 1 hour. You can keep it overnight as well, and after that we will pour out the staining solution and pour the destaining solution. Now, after the staining solution, it will stain all over the gel basically. You cannot see anything properly, but the destaining solution has the same solvent. So, basically in the Chromacid Brilliant Blue solution, what we have is glacial acetic acid, methanol and water, and then Chromacid Brilliant Blue as a powder form we just add it and make the staining solution. The destaining solution will have the ethanol and acetic acid and water, and the methanol basically acetic acid and water, but it will not have the staining solution because we want to avoid. So, any kind of non-specific staining of the gel will be avoided, and you will only be able to see your protein, and the whole gel will be white. So, now you can see there is the whole gel is stained most of the time, and you will not be able to justify or basically make sure where your protein is coming. So, for that we will keep it for some time and then we will pour it in a distilling solution. So, our staining and distilling of the gel has been done. So, we kept the staining solution for almost 1 hour and then we kept the then we removed the staining solution and put in distilling solution the gel and kept it until the background gel becomes clear and we can only able to see the protein part. So, I have here I am showing 2 gels basically. So, as I am mentioning, so we are right now what we are doing is to isolating and purifying the protein. So, first as I mentioned, you need to check if you have your protein in the cell lysate or not. So, here I am showing a test of expression. So, in this whole system basically, so this gel has been run previously before I went for all this protein purification and isolation of this protein. So, first I need to check if my protein is there or not. So, I have this is the ladder and this is the just a normal cell pellet where I did not induce my cells to produce my protein, and after a different time point I have taken out 1 ml of culture and loaded the whole system sample. So, here you can see there are lot of proteins. So, all these blue lines denotes proteins. So, basically and there are lot of proteins, along with this lot of protein there is a particular protein that is overexpressed. So, it is a very fat big band and that is our protein which is coming around 15 kilodalton. So, we are getting that particular molecular weight from this marker. We know the molecular weight of all the bands that are present in the marker. So, this is the protein which we are trying to overexpress and this is actually present in the sample. So, after you ensure this that your protein is actually getting overexpressed, then only you go for this big culture and isolation of your protein. Now, if you see what we got after isolation also. So, first in this particular lane what we ran is, so after getting the cell pellet and dissolving the cell pellet, we went for this sonication and then centrifugation, and then we have taken only the supernatant part after the centrifugation. Now, if you load only the supernatant part, you will be seeing this there are other different proteins and as well as there are the protein which we wanted to overexpress, and this is our basically protein of interest. Along with our protein of interest, there are different small bands above and at the lower bottom as well, which are as present as a contaminant. This is a different protein, this has a lower molecular weight, this is not of our interest. Now, this is the ladder which we have loaded here along with our protein, and here you can see there are only clean bands present. So, this is all the fractions that are present in that particular peak. So, basically these are the respective fractions that are we collected from the FPLC system and this shown as a peak, and you can see that there are no other proteins that are present in the upper part or the lower part of this gel. That means, this is the only portion protein that are present in our sample and it is quite clean. That means, the protein is actually pure protein. So, from this gel you can actually see the qualitative nature of this protein. So, it is pure, and from this absorbance we also got the amount of protein we have. So, now after this whole procedure we got a quite clean protein, pure protein, as well as the amount of protein which we got. Now, everything the protein right now what is in a buffer that is present is in trace NaCl. Now, for different biological experiment or biochemical assay or even during storage, you need to have your protein in a different buffering system. So, for that you need to go for a buffer exchange or sometimes even have to go for a desalting purpose. So, right now our protein is in a very strong buffering cell, it is in trace, and also the molar concentration of salt is quite high, around 200 millimolar. Now, if you want to buffer exchange your protein, as I have shown previously, you can use a Centricon. So, basically you load your protein and then you pass, you also add some amount of the new buffer that you want to keep. Suppose in our case we are keeping it in a phosphate buffer. You load, suppose you have 1 ml of protein and then you load around 15 ml of your new buffer, the phosphate buffer, and then you concentrate out. So, that will ensure, so you have to go for twice or thrice, and that will ensure that your protein has been buffer exchange in the phosphate buffer. So, you can use this kind of instrument. This is a centricon or amicon, where you forcefully due to the centrifugal force, the buffer will go out, the right now whatever present buffer will go out, and the new buffer will come in that place, and your protein will be in a new buffer. Another way of doing this is a dialysis. So, that is another most convenient and classical way of doing desalting and the buffer exchange procedure. So, for dialysis what will happen is, in a dialysis bag, so basically it is a semi permeable bag, it is mainly made up of cellulose. Semi permeable bag, we will keep our protein, and then the whole bag will be kept in a new buffer system, which is in our case is a phosphate buffer, and when we keep it for overnight or more than 8 to 10 hours. So, due to the semi permeability of the membrane, it will ensure that any small molecules or the buffer, that the water will go out and distribute in the new buffer, and it will make a equilibrium among the outer buffer and the inner buffer. So, basically if you have a higher salt solution, suppose 200 millimolar of salt in your sample, and the outer buffer is around 50 millimolar or so, then the high concentration of salt will go out from the membrane, because it is a very small molecule, it is permeable for them. It will go out from the solvent, and it will create an equilibrium due to the diffusion of the salt, and then your protein will be in a new buffer system, and also the smaller tris and cell will go out as well. And if you have any smaller protein fragments like that, if you or smaller molecules that are below the cutoff of that particular membrane, that will also go out from the membrane, but since your protein is quite big, it will not go out. So, like this. So, using this you can actually concentrate your sample as well as change your buffer, but dialysis will only ensure that your buffer has been exchanged, but it will not go for any kind of concentration. So, here I have already made a dialysis system. So, in this particular beaker, what I have is around 800 ml of new phosphate buffer. So, this is a very low concentration of phosphate buffer, 20 millimolar of phosphate buffer, and around the salt is around 50 millimolar of NaCl. But our protein was, as you be remembering, that is in 200 millimolar of salt and in NaCl. So, now in that particular buffering system, what I have kept it here is my protein. So, you can see there is a small bag or pouch. So, this is the semi permeable membrane, basically it is also called a snake skin here. So, basically you can purchase this kind of membrane. This is a semi permeable membrane. I have cut it out in a small portion, and then I have stick this two clip to the either part of this membrane, so that my protein is inside this particular bag and it does not go out or leak is prevented. So, these are the clips basically, and the whole system. So, here inside this particularly I have around 2 ml of my protein, and I have dipped this whole thing inside the new buffer system, and there is also a magnetic stirrer here. I have kept this whole thing in a magnetic stirrer overnight. So, this will ensure that proper diffusion is happening, and the salt and other small molecules will go out from this solvent, go out from my sample, and it will create an equilibrium along with this buffering system, and my protein will be in the new buffer. So, right this way I can ensure that the desalting and the buffer exchange is happening. Now, the main when you can choose to go for dialysis, because this is a very non-harsh technique, you are not putting directly your protein in a new buffer, that might cause sometimes that might cause precipitation. So, what you are doing is you are gradually changing the buffer, that sometimes helps. Otherwise, if you have a time issue or you know your protein is quite stable in a new buffer, you can use a centricon. And even after this dialysis, if you need to concentrate your sample for different biochemical purpose or different assay, or you need to concentrate and keep it store your protein, then also you have to go for a concentrator or different concentrate your sample. After this dialysis happen, we will take out the sample and concentrate using a concentrator.